After studying this module, you will be able to know about the significance of drug-dose relationship, the types of dose-response relationship and the concept of hormesis and threshold dose responses. Dose is the most significant determining factor of toxicity. The dose is the actual amount of a chemical that enters the body. The dose received may be due to either acute or chronic exposure. An acute exposure occurs over a very short period of time, whereas chronic exposure occurs over long periods of time such as weeks, months or years. The amount of exposure and the type of toxin will determine the toxic effect. Dose response is the relationship between exposure and health effect that can be established by quantifying the response relative to a cumulative dose. This relationship is important in defining the toxicity of the exact substance. It depends on the concept that a dose or a time of exposure to a chemical, drug or toxic substance will cause a response on the exposed organism. Generally, the larger or more intense the dose, greater is the response or the effect. This is the connotation behind the statement of Paracelsus, the dose makes the poison. Assume the idea of a dose response. There have to be a dose or exposure level below which the injurious or adverse effects of a substance are not observed in a population, and that dose is referred to as the threshold dose. This dose is also referred to as the no observed adverse effect level or NOAEL, or the no effect level, that is NEL. These expressions are frequently used by toxicologists while discussing the relationship between the dose and the exposure. However, for carcinogen substances causing cancer, no safe level of exposure exists, since any exposure could result in cancer. The features of exposure and the spectrum of toxic effects congregate in a correlative relationship, as a matter of course referred to as the dose-response relationship. Whatever response is selected for evaluation, the relationship between the degree of response of the biological system and the quantity of toxicant administered assumes a form that occurs so constantly as to be considered the most fundamental and pervasive concept in toxicology. Next is significance of dose response. The dose response concept is of such fundamental importance to toxicology that it provides an outline by which toxicology may be defined. The concept of the dose response indicates the requirement to evaluate the complete dose response range from very low doses that are apparently without obvious effect to higher doses that cause direct toxic effects and mortality, and the fundamental mechanisms of these effects. Consequently, disagreeing to whatever might be largely assumed and usually shown, toxicology is not a discipline that is exclusively concerned with adverse effects. It should be concerned with all practices that affect life, including adaptive responses. Overlooking adaptive responses in the study of toxicology prejudices the aptitude to improve correct and more vital considerations of the dose response. The dose response concept is therefore one that combines long thought conceptual differences between the disciplines of toxicology and its chronological parentages of pharmacology and physiology. The dose-effect relationship is the relationship between dose and effect on the individual level. An increase in dose may increase the intensity of an effect, or a more severe effect may result. A dose-effect curve may be obtained at the level of the whole organism. the cell or the target molecule. The dose response may be affected by an extensive range of biological factors, including progressive developments, age, gender, dietary and nutritional status, genetic factors, pre-existent disease conditions, and prior exposure history, among other factors. In the case of population level dose response description, the observed response is an incorporation of multiple individual dose response relationships arising in different cell types and at different molecular sites within those cells. Refined effects that occur at low doses may be screened or overcome by more obvious responses occurring at higher doses. Next is the general theory of dose-response relationship. Numerous assumptions must be considered before dose-response relationships can be used applicably. The first is that the response is due to the chemical administered. To define the relationship between a toxic material and an observed effect or response, one must recognize with practical certainty that the relationship is actually a causal one. For some facts, it is not always obvious that the response is a result of chemical exposure. A second assumption appears simple and apparent, that is, the magnitude of the response is in fact associated to the dose, possibly because of its apparent simplicity. This assumption is often a cause of misinterpretation. It is certainly a combination of three other assumptions that persist often. Firstly, there is a molecular target site with which the chemical interacts or initiate the response. Secondly, the production of a response and the degree of response are related to the concentration of the chemical at the target site. And thirdly, the concentration at the site is in turn related to the dose administered. The third assumption in using the dose-response relationship is that there exists both a measurable method of measuring and the detailed means of expressing the toxicity. For any given dose-response relationship, a pronounced range of conditions or endpoints of toxicity could be used. The best standard would be one closely associated with the molecular events resulting from exposure to the toxicant. It follows from this that a given chemical may have a family of dose-response relationships, one for each toxic endpoint. There is an assumption that the effects of a chemical as a result of exposure to a particular dose are certainly followed by a justification of the pathway by which that dose stimulated the response. This relationship has conventionally been known as the dose-response relationship. The result of exposure to the dose is any measurable, quantifiable or noticeable indicator. The response depends on the quantity and route of chemical exposure or administration within a given period. From a practical viewpoint, there are two categories of dose-response relationships. First is the individual dose-response relationship, which refer to the response of an individual organism to variable doses of a chemical, often mentioned as a graded response because the measured effect is continuous over a range of doses. And second is the quantile dose response relationship, which illustrates the distribution of individual responses to different doses in a population of individual organisms. It is also important to distinguish that a given chemical may have multiple different molecular, biochemical and cellular effects, each with its own dose-response relationship. As a result, the nature of a toxic response might very well be different at low doses than at higher doses. Next is graded dose response. Graded dose response is also known as individual dose response. The graded dose response defines the relationship of an individual test subject or system to increasing or continuous doses of a chemical. The concentration of the chemical is inversely proportional to the number of surviving cells in the cell culture system. In certain fields of toxicology, particularly mechanistic and in vitro systems, the term dose response is more accurately referred to as concentration effect. This change in terminology makes note of the specific effect on a measurable factor that corresponds to a particular plasma concentration. Graded dose response relationships are characterized by a dose-related increase in the severity of the response. The dose affinity of the response often results from an alteration of a specific biochemical process. Next is quantal dose response. In contrast to the graded or continuous scale dose response relationship that occurs in individuals, the dose response relationships in a population are by definition quantal or all or none in nature. That is, at any given dose, an individual in the population is classified as either a responder or a non-responder. Although these distinctions of quantal population and graded individual dose-response relationships are useful, the two types of responses are conceptually identical. A typical quantal dose response curve is illustrated by LD50, that is lethal dose 50% distribution. The LD50 is a statistically calculated dose of a chemical that causes death in 50% of the animals tested. The doses administered are also continuous or at different levels, and the response is generally mortality, gross injury, tumor formation, or some other criteria by which a standard deviation or cutoff value can be determined. In fact, other conclusive factors such as therapeutic dose or toxic dose is determined using quantal dose response curves, from which are derived the ED50, that is effective dose 50%, and the TD50, that is toxic dose 50% respectively. Next is threshold dose response relationship. Threshold dose response relationship is a dose response relationship that assumes that adverse effects occur only when a threshold dose is exceeded. An essential feature of the dose response relationship at low doses is the concept of the threshold. The marginally effective dose of any chemical that induces a stated all or none response is called the threshold dose, even though it cannot be determined experimentally. The threshold dose is defined as the lowest dose of the chemical that would be expected to stimulate a response under conditions at which the analysis was performed. The threshold dose is often empirically assessed as being a dose less than that the lowest dose at which an effect was measured, but higher than the greatest dose at which no effect was detected. It has long been recognized that acute toxicological responses are associated with thresholds. That is, there is some dose below which the probability of an individual responding is 0. Obviously, the identification of a threshold depends on the particular response that is measured, the sensitivity of the measurement, and the number of subjects studied. For the individual dose response relationship, thresholds for most toxic effects indeed exist. Although inter-individual variability in response and qualitative changes in response pattern with dose make it difficult to establish a true no effects threshold for any chemical. The biological foundation of thresholds for acute responses is finely proven and frequently can be validated on the basis of mechanistic information. The threshold became the standard dose response model for industrial, chemical, drugs and radiation health effects evaluation. Next is the concept of hormesis. Hormesis is defined as an overcompensatory response to some disturbance in homeostasis. Thus, hormesis is characteristically evident at low doses of a chemical at which gross disturbances in homeostasis do not disguise the hormetic response. Further, hormesis normally presents as an effect opposite to that stimulated at higher levels of the chemical. There is substantial evidence to suggest that some non-nutritional toxic substances may also impart beneficial or stimulatory effects at low doses, but that at higher doses they create adverse effects. This concept of hormesis was first described for radiation effects but may also pertain to most chemical responses. Thus, in plotting dose versus response over a wide range of doses, the effects of hormesis may also result in a U-shaped dose-response curve. In its original development, the concept of hormesis pertained to the ability of substances to stimulate biological systems at low doses, but to inhibit them at high doses. The application of the concept of hormesis to whole animal toxicological dose-response relationships may also be relevant, but requires that the response on the ordinate be variant with the dose. A present renaissance in interest among pharmacologists in low-level effects stems from use of homeopathic methods to treating disease. Promoters of homeopathy retain that low levels of toxic materials stimulate physiological responses that can target disease without producing adverse effects in the individual undergoing treatment. Homeopathic principles may have application in toxicology based on the basis that exposure to some chemicals at sub-threshold levels as defined by standard acute toxicity evaluations can cause toxicological as well as pharmacological effects. Both pharmacological and toxicological homeopathy may be the consequence of hormesis. We will conclude this module with summary. Dose is the most essential determining factor of toxicity. The dose is the actual amount of a chemical that enters the body. The dose received may be due to either acute or due to chronic exposure. Dose response is an association between exposure and health effect that can be established by defining the response relative to an increasing dose. This relationship is important in defining the toxicity of a particular substance. The concept of the dose response implies the need to assess the entire dose response continuum from very low doses that are seemingly without demonstrable effect to higher doses that cause frank toxic effects and mortality, and the underlying mechanism of these effects. The dose-effect relationship is the relationship between the dose and effect on the individual level. An increase in dose may increase the intensity of an effect, or a more severe effect may result into the death of an individual. The dose response may be affected by a wide range of biological factors, including developmental processes, age, gender, dietary and nutritional status, genetic factors, pre-existing disease conditions, and prior exposure history among the other factors. From a practical perspective, there are two types of dose-response relationships. First is the individual dose-response relationship, which describes the response of an individual organism to varying doses of a chemical, often referred to as graded response, and the second is the quantal dose-response relationship. For many chemicals and effects, there will be a dose below which no effect or response is observed. This is known as the threshold dose. And there is a concept of hormesis, which is defined as an overcompensatory response to some disturbance in homeostasis. Thus, hormesis is characteristically evident at low doses of a chemical at which gross disturbances in homeostasis do not disguise the hormetic response.